Welcome back to Microbiology Lab. This is Lab Practical 1, Day 3. We're looking at, we're going to look back for just a quick moment at the negative stain because I forgot to show you the results from that. And then we're going to move forward with a capsule stain. So let's take a quick look at the syllabus. I always like to make sure that we've checked off all of our topics for the agenda. So to remind everyone of where we are. The syllabus shows us here for September 1st, and so we have less to do than some days. The capsule stain, exercise 3-9, and then it says check and discuss environmental plates and observe bacterial colony types and fungal types as well. So we're going to be taking a second look back at the sample that I took from the bottom of my shoe, as well as the sample, what was the other sample? It was the shoe, let me check here. My shoe, and my cell phone. And then also the lab air. And so we'll be looking at those things, those items. And now it's time to resume, starting our coverage of today's activities. And we'll start by looking back at the negative stain. So I introduced the negative stain, and if you recall from last lab activity period, I made a negative stain right here, and I didn't put it under the microscope to show you guys. So what I'm going to do is take you in for a close-up view. We'll take the camera and we'll see what came out of this negative stain. However, one of the things that before I take a look at that, I want to remind you guys of what you're looking for. So you'll likely see a lot of ordinary bacteria that appear clear, colorless, because they repel the dye in the negative stain, and then the background will have the dark color. So you can have some contrast to see the bacteria. Keep in mind that the bacteria are going to be either rod-shaped or spherical, so they'll have fairly regular looking shapes. Occasionally though, if you get lucky or you find something a bit unusual, you might end up seeing these rarer spirochetes that sometimes appear in people's oral cavities in a negative stain sample like this. We talked about some dangerous spirochetes like Treponema pallidum, which is the cause of syphilis, and we talked a little bit about Lyme disease, which is caused by an organism, Borrelia burgdorferi. And not all spirochetes are guaranteed problematic. So to give an example, Treponema denticola is an organism that can look like this under the microscope, like little squiggly, wavy lines, and they do come from people's gum line, or where the teeth meet together, and it provides an environment where sometimes little Treponema non-pathogenic bacteria can be found. I have read articles that say that when people have a lot of tooth decay or gingivitis, complicated dental oral problems, sometimes Treponema denticola, the oral spirochetes, can actually increase in abundance or be more likely to be found in those samples. But at the same time, we haven't seen that Treponema found in the oral cavity is a direct cause of oral pathology. So there's a correlation, but the causation isn't known as far as I understand from the research so far. So I'm going to go ahead and pause the camera and show you guys a picture of a negative stain through the microscope. Not a picture, but a view through the microscope. All right, so we're taking our look through the microscope at a view of a negative stain taken from an oral cavity. And depending on if you can see this through the camera, at the end of the pointer, there's the black needle-like device in the field of view here. I can see a spirochete as just a wavy little snake-like line. And I can actually see more than one spirochete in this particular field of view.
if you look around in here, you change your focus and you move the slide around, you can see through some fine focusing and some careful observation that there are multiple spirochetes. I confess that this is not mine, and part of the reason is because I had looked through it moments ago, and I didn't find anything as interesting as a spirochete, and so what I wanted to do was just take the time to show you. This is from a student some years ago. I know I had told that story about some of my students having found them, and they, and they are findable occasionally in people's oral cavity. Doesn't necessarily mean disease. So here we have a spirochete at the end of the pointer, and so you got a good look at that. This is likely the type of image that you would see on a practical exam, or in the case of, if you were a clinician and you were looking at someone's clinical sample, this could be an important finding.
So now that we've looked at spirochetes, we're going to move forward to the capsule stain procedure. Just to remind you, the spirochetes that we saw were on against a red background because we used Congo red as an acidic dye appropriate for the negative staining technique. This was one that was used with India ink as an alternative to the Congo red that we used. Either one works fine as long as you have a dark enough background to see the clear little cells. So we saw wavy lines like this that indicated spirochetes in the sample. Now that we're going to be looking at the capsule stain, this is a good segue because the capsule stain reveals something different about bacteria other than just their shape. It reveals this structure that we're going to learn about called the capsule, but the capsule stain procedure stems from using a negative stain beginning step and then modifying it from there. And so I have saved my negative stain so that I can actually use my negative stain to make a capsule stain. And so we'll see how that works. Okay, for the capsule staining activity, we are going to use this staining to differentiate between cells that produce a capsule, like we see here with a clear, indicated by a clear zone around the bacterial cells, and then those that do not produce a capsule. So they're a little harder to see, but within this field of view, you can see that there are these dark pink little cells here and there, not too many of them, but some of them lack the capsule, and maybe they're younger or they haven't had a chance to make a capsule yet, but the ones that have a capsule have these clear zones around them that are unstained, and they jump out as pretty noticeable when we do this procedure. You would not be able to see encapsulated bacteria nearly as readily if you were using an ordinary staining technique like even the negative stain itself. Without modifying it, wouldn't show you these capsules and their presence as readily. So simple staining techniques, negative staining techniques are not sufficient, so we go to the capsule stain. Before we start that procedure, I'd like to just talk about what the importance is. The function of the capsule is something that acts as a layer of protection for bacteria. Not all bacteria can make a capsule, so this is unique to only certain bacteria, but some of the ones that make capsules are really medically difficult to treat because that layer of protection can make them more resistant to things like antibiotics and immune response. So let's look at the functions of the capsule. First and foremost, the capsule works as an extra layer of protection. So it protects from various factors that could be threatening to bacterial cell lives. And let's think about what those things could be. So I had mentioned that antibiotics, which we know is almost always the treatment for bacterial infections, they may be less effective. And that's because these bacteria, when they have capsules, they're also sticky. They can stick to one another, they can stick to their environment, and they can form layers of growth that then acts as a physical shield so that it's more difficult for antibiotics to get in. When those layers form, they're usually called biofilms. Biofilms are then very difficult, because in a disease process, you've got layer upon layer that the antibiotics are not able to penetrate into to effectively kill the bacteria. So we can say antibiotics, but we can also throw in other antibacterial products like, or tools like antiseptics, disinfectants, and whatever else you can name that you might use to try to control bacteria. So I'm going to go ahead and write all of these down. Protects from antibiotics, disinfectants, antiseptics. Long words, not a lot of space to fit this in, but you should jot that down. Also, the capsule acts as a physical protective layer from just ordinary elements. So when a bacterial cell finds itself in an environment where maybe there's not enough moisture, so fluid loss or dehydration can be difficult for bacteria. It can threaten their lives, or just simply put them in a situation where they're stuck until they get more moisture. The capsule has moisture within it. Capsules are actually made of chemicals that are either carbohydrate based or polypeptide based, which would be protein based. So either sugars or proteins make up the capsular structure, and these capsules then protect from dehydration. But because capsules are also made from sugar-based compounds or protein-based compounds, they can also act as an extracellular form of food storage. So a big fat capsule like we see around this particular organism here in this rod-shaped bacterium in the picture can grow quite large under ideal conditions, even bigger than the volume of the bacterial cell itself. So capsules can get gigantic, but then under a period of starvation or dehydration, this organism can start to actually contract its capsule and bring those bits of food and moisture back into the organism under tougher times. So we can say that when they're well fed, they're fat and happy, and when they're under stress, the capsules may not be made or can shrink. So capsules are also beneficial to the bacteria that can make them by protecting from dehydration and starvation. We should bring in a third item as well that the capsule would be able to protect bacteria from. Capsules also protect bacteria from I'm forgetting. What's the third thing? Give me a moment. How can I forget? The immune system. Our biggest defense against bacteria in general, and even these encapsulated ones, would be our immune defense. So let's take into account where encapsulated bacteria might cause disease. These organisms are described as having capsular structures that are not only just sugary or protein-based, polypeptide-based, but we say that they're mucoid. Mucoid means mucus-like, and we know that mucus has a sticky quality to it, and so this picture is of an organism called Klebsiella pneumoniae, and that implies that it infects the lungs. I'm sure most of you have heard of pneumonia. This just medically means fluid in the lungs. And there are many reasons or causes for pneumonia, but one of the most common causes for pneumonia are bacteria in the lungs that definitely don't belong there. The lungs themselves are almost sterile. They're constantly being patrolled by phagocytes, which are white blood cells that police the area, especially down in the lower respiratory tract where the lungs have little air sacs that exchange oxygen and carbon dioxide and regulate our blood gas. But in that vital area, deep within our lungs, there are these phagocytes that phago means to eat, and cyte means cell. And these are white blood cells that will eat little bacterial cells and fungal spores and viruses and whatever the phagocytes can get a hold of. And the ones that are specialized that live in the air sacs called alveoli, those alveolar air sacs have the phagocytes called macrophages. So alveolar macrophages are the white blood cells that would normally eliminate these kinds of Klebsiella pneumoniae cells. Interestingly, Klebsiella pneumoniae can live more commonly in the gut. So people can have this inside of their intestines. It can be in the digestive tract, hanging out, doing something that we wouldn't notice, just growing and multiplying or colonizing in a non-disease causing way. But then these organisms, Klebsiella, can get out of the gut, and sometimes they stick with their mucoid polysaccharides in the urethra and cause urinary tract infections. So a person can get UTI from Klebsiella pneumoniae, or as its name implies, it can also get in the lungs. And so we can say that one of the reasons why they cause disease is because the phagocytes, the alveolar macrophages or other phagocytic white blood cells, have a difficult time trying to ingest and perhaps even digest these particular encapsulated cells. They're stuck to the lung tissue or the urinary tract mucous membranes, and then the phagocytes come along and they're trying to acquire the encapsulated organisms, and the organisms are not coming up easily because they're not ordinary bacteria. They're stuck in these layers, and so they're described very accurately as, the capsules are described accurately as anti-phagocytic. And now you know why. You fill in the blank. Another thing you could say about anti-phagocytic capability is that the capsule may provide a covering that makes it difficult for phagocytes to recognize the cell itself. So if a phagocyte, white blood cell, comes along and is trying to recognize that there are bad bacteria, they may have more difficult time recognizing, acquiring, and ingesting, or even perhaps digesting these disease-causing types of bacteria. We can also say that the capsule is simply an adherence factor. It helps the cell, these cells, to stick down to their environment and colonize sometimes parts of the body that a disease process starts up in that. So we'll just say they are also considered an adherence factor. Another word that we can use to refer to an adherence factor is to call it an adhesin. And you'll hear these terms come up in lecture. So one such adherence factor is to say adhesin, and the capsule is an adhesin. Bacillus anthracis was mentioned just a little bit in one of our first days of lab and lecture, talking about the genus and species of anthrax. Anthrax can get into the lungs, and so pulmonary anthrax is one of the deadliest types of infection, where rod-shaped bacteria can make spores that are in the soil or in the hide of an animal like a cow or sheep. Sometimes Bacillus anthracis lung infection is referred to as wool sorter's disease. And what happens is people inhale Bacillus anthracis spores, and then they germinate, they grow and they multiply, and immediately they try to start making these capsules and stick to the lung tissue and evade the phagocytes, and they defeat the phagocytes in more than one way. Bacillus anthracis can actually secrete something called lethal factor, LF, and so lethal factor, as the name implies, would be a chemical that kills phagocytes. And not only can they secrete this killer lethal factor, but they can also secrete another chemical called edema factor. So LF and EF, edema, E-D-E-M-A. Edema means swelling, and so edema factor is referring to the fact that when Bacillus anthracis gets in, this chemical that they secrete causes a lot of inflammation. And when inflammation occurs in the lungs, that contributes to fluid buildup. All this tissue fluid, pleural effusions, build up inside the lungs, and a tidal wave of moisture and liquid stops gas exchange. And so people can die within a couple of days of a pulmonary anthrax onset. So a person can go from stable and seemingly okay to dead within a handful of days. And what's crazy is that Bacillus anthracis not only has this capsule, which is able to cause these secretions to occur, but also the capsule itself is made of a mucoid polypeptide. So rather than a mucoid polysaccharide like most of the common capsules that we study, this pathogen has a polypeptide, which is a protein-based type structure, and it's not digestible by normal events like, say, a phagocyte digesting and trying to, or ingesting and then trying to digest the capsule. So the capsule itself becomes this undigestible shield, and that's why Bacillus anthracis is just so bad. So amazingly, if someone does become the victim of say, Bacillus anthracis lung infection, the pulmonary anthrax event can be treated with antibiotics. So ciprofloxacin, for instance, a broad-spectrum antibiotic, if given soon enough, could actually cure a case of pulmonary anthrax before it would become deadly. But otherwise, if left untreated, this is almost certainly a deadly event. So there are many different what are called virulence factors. Virulence means disease-causing factors, and the capsule is one such virulence factor, also known as an adherence factor or an adhesin. So we're learning a lot of new terms here, but we want to make sure that we understand this term virulence as well, since a capsule is a virulence factor. Virulence will define as disease causing. Okay, that's a thorough description of capsules and how they apply, and now how they apply to understanding bacterial disease in some cases. And we need to discover how, in fact, those capsules are present in not so much how, but I mean what they look like when they're present. And so we're going to go out of our way to do a capsule stain. All right. Here are all the different steps of the capsule stain. And what I would like to do is take a close-up view so that you can see me apply the different steps. Before I do that close-up, we have already done step 1, which was to use a half of a drop of Congo red, mix in the bacteria, and make a smear using a second slide at a 45-degree angle. And this first step is to make a negative stain, essentially, and then let it air dry. So I have a negative stain at this point. This was from my oral cavity from last activity. You could actually do this activity with Klebsiella pneumoniae. Shockingly, we actually are able to safely handle that in here, as bad as it sounds. Aerosols are highly unlikely coming from a test tube and onto a slide, rather than having them coming out of the lungs of a person who's infected. So we have Klebsiella pneumoniae that I can show you under the microscope. I have some pre-prepared slides. We also have Bacillus megaterium available during some classroom meetings. And just another organism. Bacillus megaterium happens to be a non-pathogen that just lives in the soil and happens to have capsules in common with, say, like its cousin Bacillus, well, not literally a cousin, but a related species like Bacillus anthracis. So we can look at Bacillus megaterium, we can look at Klebsiella pneumoniae as model organisms for the capsule. So I will pause it, and then I'll be showing you a close-up of how to take care of the rest of the steps remaining here. what I'm going to be doing is gathering up these other chemicals like hydrochloric acid and safranin. All right, as we take in a zoomed in close look, I have my negative stain. Now, since I'm actually going to use this for my capsule stain, one of the things I can do is remove this label, or just put some alcohol on there, and I can take that label off of there, or I can modify it to show that it's also, to label it as a capsule stain. So I've got a fully air dried negative stain, and if you recall, I used Congo red, and that's what you would use for the first step of this capsule stain. And now that's fully dry on the slide. And the next step in the capsule stain after air drying that without the use of heat is to use 0.1 normal HCl. Basically, this is a hydrochloric acid solution that's strong enough to kill the bacteria and also to fix them to the slide so that they don't just rehydrate and wash off. Now, I'm going to go ahead and flood the whole slide with hydrochloric acid. And so you want to make sure that you get all the bacteria, so they're all dead, and that it fully covers the slide so that it changes its color as well. You'll actually see that because of the dramatic pH change that hydrochloric acid produces as it reduces the pH to almost 1, that the Congo red turns into a midnight blue. Now, I'm going to look at the clock and make sure that that's going to sit on there for about 30 seconds. You want to give the hydrochloric acid enough time to act, but not so much time that it completely washes away your dried negative stain. So I'm waiting the full 30 seconds, and during that time, I'm going to use a clothespin or whatever other device you can use for a slide clip. I don't want to get the hydrochloric acid on my fingers. That would burn my skin. I don't want to see anyone burn their skin or their hands. If you did get this on your skin, you could just wash it off right away. It's not a super strong solution, but I'm going to go ahead and dump off the hydrochloric acid and try to shake off any excess. Now you can see that the hydrochloric acid did take up some of the dye, and it looks kind of spotty or patchy now, and we're not going to be able to rinse that. If you rinse that, what happens is it can wash off even more. So this is really delicate staining process, but it did turn out well so far. We're going to follow up with this last step of an application of a red dye called safranin. You would not want to mix up the two red dyes. Congo red is always the first step when you perform the negative stain first step, and then you put that aside. You want to make sure you grab this second type of red dye. It's a kind of a little bit different shade of red, kind of a light reddish pink. And what we're going to do is flood the entire smear. Be generous with these chemicals, guys. You don't want to hold off and put too little. So you cover the whole smear, and then you note what time it is on the clock. We need to let that soak for 1 minute. And I'll explain what these chemicals, what they accomplished in detail in just a moment after the close-up view. So while we're waiting for that 1 minute, you have to have your slide clip ready to go. We want to be careful about the timing and not leave these chemicals on for too long. And we can get ready to rinse. We're going to do a light rinse, and the amount of water that you're using, because this is really delicate, it's going to be just the softest stream of water that you can provide so that you can get the safranin off of there, and then you can blot dry your slide. Okay, I'm going to go ahead. It's been approximately 1 minute. I'm going to dump off the excess safranin and then give it just a very gentle rinse. And notice the way that I rinse. I'm rinsing with the slide at an angle, and I'm using my slide clip so I don't get these chemicals on my fingers. And that's it. So now I'll show you the blotting technique. You can use a paper towel or a Kim wipe. I've got a Kim wipe here, which is like a delicate little towelette. And then what I'm going to do is just
wipe the bottom. It's okay to wipe the bottom of the slide because the bottom has nothing on it. It's just bare glass. The top you wouldn't want to wipe because then you'll damage the smear. So what I'm going to do is take my Kim wipe and just press it down gently and blot it. Maybe I'll do that one more time, and that's about as much as I want to do. Any little bit of leftover moisture is not a problem. This can go under the microscope. No cover slip. You can just go straight to viewing, and you can put oil on there and everything. No problem at all. So that's the capsule stain now completed. All right, so looking back at the steps, we want to take note of a couple of important points, taking into consideration what the chemicals actually do. So the first chemical was Congo red, which we know allows us to make the negative stain technique. And then after letting it air dry so as not to use any heat, let's think about that. Capsules are actually heat sensitive. So if you were to use some sort of heating mechanism like a slide warmer or a flame, it would shrink these capsules, and effectively it would make for a bad capsule stain. It might ruin the capsules. So we don't use heat. We have to be patient about the air drying. The next thing we have is the hydrochloric acid. And so I'll underline this. I've already typed it in. Hydrochloric acid kills the bacteria and coagulates the proteins. And so protein coagulation is the way in which they die. And killing the bacteria is important because you want to make sure that those bacteria, especially if they were pathogens, like in a clinical sample, that they're not alive anymore. And so that's an important, that's something that we have to have ways of making our samples safe, and understanding also that what this does is the hydrochloric acid sticks the bacteria to the slide. When the proteins coagulate, it also causes them to fix down onto the slide. Now, the last step was the safranin, which was a 1-minute procedure followed by a gentle rinse. So the safranin has a different function than the Congo red, because even though safranin is a reddish pink dye, it's going in directly to the bacterial cells. Recall that on the negative stain, the whole background is colored, but the cells are clear. In this case, we see that the capsule stain, the cells are not clear. You can see the little bacterial cells inside of each and every one of the capsules, and that's because the safranin goes in and actually gets absorbed by the cells. And if you review back through your notes so far, the negative stain has a negative charge, and it's repelled by the bacterial cells, so it stains the background but not the cells. Safranin, on the other hand, is one of the simple stains, one of the basic alkaline dyes, and if you recall, that has the opposite charge. It has a positive charge, and so the negatively charged bacterial cells will absorb the positively charged safranin, and then you'll have this contrast between the stained background and the stained bacterial cells, and then the unstained capsule. And you might be left wondering, well, how does the capsule not take on the stain? And apparently, capsular structures, which are made of these mucoid polysaccharide or mucoid polypeptides, they lack a charge. And without a charge, then the negatively or positively charged dyes don't really interact with the capsule. Okay, so now it's time to pause one more time, and we're going to look at capsule stain result through the microscope. And remember, we're looking for something that's going to look similar to what we see in the picture here. Okay, now we're looking through the microscope lens at low power. And I have the camera positioned here. I'm going to go ahead and use a little fine focusing to try to get them in sharp focus under low power. And what I want you to pay attention to under low power is not so much that you can see the bacteria, but the color. So on the left, we see that there's this dark bluish, purplish color, and that's from the pH change when I was describing that the hydrochloric acid would cause the Congo red to sometimes turn like a dark blue or almost midnight type color. But then we can also see parts of the smear here that are still the red color of Congo red. And it really depends on what part of the smear is either thick or thin and how much pH change happened from the hydrochloric acid. Regardless, it doesn't really matter when it comes to seeing capsules. So don't be confused if you see either red or kind of a blue color, because either background color will work to give us that contrast, and then the bacterial cells, when we take a closer look, are still going to have the reddish pink color from the safranin. So what I'm doing is putting just a little bit of oil. I know you can't see anything right now, guys, but I'm putting a little bit of oil, and I'm going to carefully switch to the oil lens magnification and do a little fine focusing, and there we are. You can see all the tiny little bacterial cells. This is a slide that was labeled Klebsiella pneumoniae, and you can see that all the little clear spots are where the bacteria have capsules around them. And then inside of those little clear spots, it's maybe a little hard to zoom. I'm going to try to zoom in. We can see by zooming in that there are bacteria that are colored with the safranin. And that's about as good of a close of a look as we're going to get on the camera through the microscope.
So there you have it. We saw a microscopic view, and we got to compare it to the picture taken from your lab manual of the capsule stain. And that's as far as we're going to take that particular activity. And we're going to now take a look back. So one last time, we're going to look back at the environmental samples that we all looked at from the first day, from the bottom of my shoe and the microscope. And so there are various types of colony specimens or appearances that the bacteria can take on as they grow on the plate further into maturity. Generally speaking, what we're going to be observing in just a moment is that the colors will get a little bit brighter or more vibrant because pigments have had a chance to build up more, colonies have had a chance to get bigger, and also importantly, fungi, which are slower growing types of organisms that are different than bacteria, will have had a chance to actually grow where bacteria may have grown already very quickly. And just as a quick note, since I caught it out of the corner of my eye, we were talking about Klebsiella pneumoniae during the capsule stain. You saw capsules under the microscope just now, but check them out on a plate. So here are the mucoid, round, raised colonies of Klebsiella pneumoniae growing on a Petri plate. So when people have Klebsiella pneumoniae lung infection or a urinary tract infection, if you take the sputum, which is a mucus sample coughed up out of the lungs, or you take a urine sample from someone who has UTI, if you were to take a sample of that and spread it around on a Petri plate, these would be the kind of colonies that you would see growing to have this appearance. And then you could do your capsule stain by taking a little sample from one of these colonies. You could take a loop and flame it to make sure it's nice and sterile, and then grab a little bit of one of these colonies and put it through the capsule stain process, and you can verify that in fact the bacteria do have the capsules. And then with a few other techniques that we're going to learn, you could then confirm that it's actually Klebsiella pneumoniae and narrow it down to the genus and species level. So I'll put the document camera on, and then we'll compare the pictures, or what you guys saw the first time and this time. So the document camera will turn on in just a short minute. Here we are. And what are we seeing here? This is labeled my shoe. So it looks to be about the same as before, but the colonies are larger. And I was mentioning as well that the pigments are much brighter. So these were more of a light pale yellow, and now these colonies, they could be Staph aureus, or they could be another common yellow organism that comes from the skin called Micrococcus luteus. And we're not going to know because we're not going to do the testing on them, and I'm not going to open the plates, but we can see that the colonies have gotten larger and a little bit brighter in color. If we look very closely on this plate, we might also be able to see fungi. Now, how are you going to identify fungi? Most of what's on here is still bacterial, but the reason I'm judging it that way is based on colony appearance as being smooth and mostly shiny and reflective, or even mucousy looking, and very regular in appearance for the most part. Fungi tend to be fuzzy looking. Fuzzy if they're molds. And so molds have these elements called hyphae that grow up into the air, and we're going to do a fungi lab later on to take a closer look. But what we can do in this case is flip the plate over and see if maybe something on this plate looks fuzzy. So I'm trying to zoom in as far as I can, see if any of these colonies actually appear to have a fuzzy appearance. And it's a little tough to tell on camera, but not really. I mean, there aren't really many that look fuzzy. That particular one there might be a mold. It has some striations, and it's a little tough to tell, but I suspect that that's fungi. And the way that I could tell you if that's fungi for sure is to put a sample of it under the microscope. Right now we're just going on colony appearance. These look like they might be a little fuzzy to the eye, but that's typically how bacillus species look when they're taken out of the soil and grown on a plate. So not everything that's fuzzy looking is always fungal. It could still be bacterial. Let's take a look at the cell phone. Were there any fungi on my cell phone? So I'm zooming back out for just a quick moment. We'll let that autofocus. I'm not seeing anything that's necessarily showing me bacteria, but earlier I did glance at this, and if we get a different angle of this colony here, this one might be fuzzy. I'm not sure. Let's take a closer look and zoom in on that. Can we get that to focus? A little hard to tell. Okay. Well, this might be a fungus. I was talking about this before, that this colony, as irregular as it looks, does not appear to be bacillus. It's a little too erose or unusual and irregular, and it is waxy looking. And although it doesn't look like a velvety or fuzzy appearance, the waxy appearance of it is typical of something called trichoderma, and you don't have to memorize that, but again, a sample would tell us the story microscopically. Well, that's too bad we didn't have any fungi that were fuzzy to look at, but those were our two plates. Just looking back at them, they're more mature. They've had a chance to grow at room temperature for the last 5 days outside the incubator, and I think that's as much mileage as we're going to get. And I say that because our air sample grew nothing, and that is unusual. I think the micro lab has just been undisturbed for quite a while, and they had actually redone the floors so that they're nice and shiny and they don't have much dirt on them, and there just wasn't much flying through the air. And so our air sample just didn't show anything. But the air can sometimes, if you leave an air sample out for long enough, it could have a good number of the colonies that popped up from these soil samples, especially if you leave a plate outdoors. So we didn't have that to look at this semester, but at least we got a chance to see how this technique is performed And we're going to be doing study of fungi in the future, so we'll get a chance to see fungi. And that's about as much as I have for you guys today. So next time we'll be looking at Lab Practical 1, day number 4, and this syllabus. Look ahead and see which kinds of activities are listed there, and so I'll see you guys in a couple of days.